Hello my dear students, welcome to the MS Science Academy. Myself Menu Srivastav, and today we are going to discuss about the electron transport chain and its mechanism. First of all, some students, I think some of having the confusion, what is the reason behind electron transport chain? I think they very well known about the glycolysis. They can easily understood about the glycolysis, the Krebs cycle, the beta oxidation of fatty acid. All the things they can easily understand, but they can't get the relation between the electron transport chain and its importance. So, what is the reason behind the electron transport chain? First of all, either the carbohydrate, the protons, so the proteins, the lipid are metabolized into carbohydrate metabolized into the monosaccharide, carbohydrate metabolized into the monosaccharide, protein metabolized into the amino acid, and fat metabolized into the fatty acids and glycerol. Fat or lipid metabolized into the fatty acid, and these all carbohydrate, protein, fat are anyhow mixed with the TCA cycle or or interact with the TCA cycle either via conversion of acetyl coenzyme A or by forming the intermediate of TCA cycle. Okay, so either the carbohydrate, protein or lipid, whatever things which we are eating, they have to enter in the TCA cycle either via acetyl coenzyme A or via we can say the intermediates of the TCA cycle. Anyhow, they will enter into the TCA cycle, and by the TCA cycle they form the energy particles, that is the NADH and FADH2 and ATP also. Okay, ATP also. But the main thing is that the ATP is the ready-made material for our cell. Ready-made material means the cell can easily utilize the ATP and cell can get the energy. But it's not same for the NADH and FADH2. In NADH and FADH2, they are the not ready to make ATP. They are the ATP packets, but the ATP packets are just packed into or wrapped into the different forms. Okay, so from NADH, if the cell want to take the ATP from the NADH or either FADH2, then NADH and FADH2 have to go the electron transport chain. Then the energy generation means the ATP production will proceed. Okay, so now you will get the point why the electron transport chain is compulsorily. Okay, electron transport chain is the process which convert the FADH2 and the NADH into the ATP. Simple as thing. Okay, so the ATP is ready to eat. So in any cycle, if the ATP or the GTP is formed, the cell is easily take it. But the difficulty in the case of NADH and FADH2. So in that process, NADH and FADH2 is formed, then NADH and FADH2 have to go through the ETSC cycle. Then when it will go through the ETSC cycle, then they will convert into the ATP. Okay, so in the glycolysis calculation, in the Krebs cycle calculation, you will say that in one glycolysis 8 ATP is formed. Not 8 ATP directly. In that cycle, you have to calculate the NADH is equals to 1 NADH is equals to 3 ATP. So like that calculation you have to done. Okay, so that calculation NADH is equals to 3 ATP and 1 FADH2 is equals to 2 ATP. That calculation you can do only if the electron transport chain is proceed. Okay, so in this method, electron transport chain is just for the conversion of NADH and FADH2 into the ATP form. Now, the complex one, So the electron transport chain is the method to convert the NADH and FADH2 into the ATP. Okay, that's all. Now the these electron transport chain is takes place in the different enzyme process or the enzyme complex. These are the 5 complexes in the embedded in the inner membrane space. Now first we look the structure of mitochondria. There is a structure. This is the outer membrane. This is the outer membrane of mitochondria, and this is the inner membrane. Okay, this is the inner membrane, and this is the intermembrane space. This is the IMS intermembrane space, and the inner membrane has the different folds is known as the cristae. Okay, and on the cristae has the F1 particles. Okay, and that F1 particles having the some part of the electron transport chain, and in that inner membrane the electron transport chain complexes are embedded. Okay, so now you will get that where the complexes of electron transport chain are embedded in the inner membrane. There are the 5 complexes. Complexes means they are having the different kinds of the electron carrier, different kinds of the enzyme, and such kind of the packet they are having. So this is known as the complex. So there are the 5 complex 1, 2, 3, 4 and 5. We have to represent in the Roman numerals, and now we will discuss the names of the complexes. Okay, so now just take a screenshot, then I will rub it. Okay, now the complex 1 is known as the NADH. The complex 1 is known as the NADH coenzyme Q reductase. Complex 1 is known as the NADH coenzyme Q reductase. Okay, coenzyme Q reductase. NADH coenzyme Q reductase. And the other name is the NADH ubiquinone oxidoreductase. NADH ubiquinone oxidoreductase. Okay. Now the complex 2. The complex 2 is the succinate dehydrogenase or succinate coenzyme Q reductase. Complex 2, succinate dehydrogenase and succinate coenzyme Q reductase. Succinate coenzyme Q reductase. Okay. Now the complex third is the cytochrome reductase. Cytochrome reductase. Okay. And the second name is the coenzyme Q cytochrome C oxidoreductase. Coenzyme Q cytochrome C oxidoreductase. Now the complex 4, that is the cytochrome C oxidase. Complex 4 cytochrome C oxidase. And the complex 5 is the ATP synthase. These are the 5 complexes. Okay, these are the 5 complexes. Take the screenshot. Okay, now in the complexes, all the complexes will participate, and by the participation they will transfer the high energy electron. Okay, and during the transfer of high electron, these electrons will generate a proton gradient. How is it possible? We will see. Okay, these are the 5 complexes. Now the first complex is known as the NADH coenzyme Q reductase or the NADH ubiquinone oxidoreductase. This NADH will generate by the substrate. The main confusion is that what is the substrate? The substrate is that chemical only or that particular ingredient only where or in the reaction the NADH is generated. Okay, the substrate, in the substrate, the reaction is in the either the glycolysis, either in the Krebs cycle, in the beta oxidation fatty acid, where the NADH is generated in the reaction, that particular place or particular chemical or particular substance, because biomolecule is known as the substrate for NADH or FADH2. Now we will write the substrate for NADH and FADH2. Just I am going to rub it. Okay, just take the screenshot. Substrate. Now we are going to substrate. What is now you know that what is the substrate? Substrate at the particular chemicals where, or the particular reaction where the NADH and FADH2 is generated. So in the substrate for NADH, okay, substrate, I represent the substrate S for NADH, glyceraldehyde 3-phosphate, pyruvate, pyruvate, isocitrate, alpha ketoglutarate, alpha ketoglutarate, malate, okay, and the beta hydroxy acyl coenzyme A, beta hydroxy acyl coenzyme A. Okay, so these are the substrate for NADH, and substrate for FADH2, substrate for FADH2 is that succinate fatty acyl coenzyme A, succinate fatty acyl coenzyme है, mitochondrial glycerol 3-phosphate. Okay, so these are the substrate for NADH and FADH2. Now we will come to the electron transport chain main ingredient. Okay, so here in the complex 1, I have representing the complex 1, complex 2, complex 3, complex 4 and complex 5. All the complexes are embedded into the intermembrane. This is the matrix part and this is the intermembrane space. This is the intermembrane space. Okay, this is the inner membrane and this is the matrix. Okay, now come to the NADH. When it is formed, the NADH releases 2 electrons and form the NAD, and these 2 electrons is captured by FMN. FMN is the flavin mononucleotide, and these flavin mononucleotide take this high energy 2 electrons and convert into the flavin mononucleotide hydrogen, and these 2 electrons just pumped into the iron sulfur protein, and just 2 electrons just take by the coenzyme Q. Coenzyme Q is the ubiquinone, is also termed as the ubiquinone. And after taking the 2 electrons, this form is known as the ubiquinol. Okay, this ubiquinone is converted into the ubiquinol, and this ubiquinol is just returned back to the ubiquinone. Okay, just take the 2 electrons, convert into this ubiquinol, and just throw these 2 electrons into the cytochrome B. Now the main thing is that where is the complex 2? The complex 2 is the complex 2 is also come into the action, but the complex 1 and complex 2 both will meet into the complex 3. Okay, so complex 1 is worked separately, complex 2 works separately, and complex 3 is come into the action, and just complex 1 and complex 2 meet into the complex 3. So this is the point you have to keep in your mind, that the complex 2 will have to keep just that complex 2. What happened into complex 2? Let us see. the succinate is converted into the fumarate. The succinate is converted into the fumarate, and during this FADH is formed into the FADH2, and these two electrons is thrown back into the iron sulfur protein, and these two electrons taken by the ubiquinone, and this ubiquinone taking the two electrons convert into the ubiquinol and just return back into the ubiquinone, and just two electrons just go back to the cytochrome B, which is present in the complex third. The main thing is that the matrix is rich in the protons. Matrix is rich in the protons. Okay, so when these processes just going into the complex 1, 4 protons just okay, this reaction I will discuss later. Just complete this up to the complex 4. Now the electrons comes to the cytochrome B. The cytochrome B take the electron and just throw back to the iron sulfur protein. These electrons take by the cytochrome C1, and these electrons just take by the cytochrome C, and the cytochrome C just take the electron and reach up to the complex 4. The cytochrome C is the mobile electron carrier, This thing you have to keep in your mind, that the cytochrome C is the mobile electron carrier. What is the mobile electron carrier? The mobile electron carrier is that the cytochrome C is take one electron and jump to the copper ion, which is the A type, and this is the electron will just reach to the, or just given to the copper ion. Again it come back and take the second electron, your first electron take the second electron, and then again go into the copper ion and give it to the copper ion A type. Okay, so that's why the cytochrome C is the mobile electron carrier, because at a time it will take the single electron and just reach to the complex 4. Okay, and then again come back and then take the another electron and just reach to the complex 4. Okay, now the copper ion A type, A type, B type, we will not go into the in deep, but because the it's not main thing. The main thing is that in the complex 4, the two types of the copper ion A type and copper ion B type also present. The copper ion just take its electron and give it to the cytochrome A, then to the electron is just transferred to the cytochrome B type and the cytochrome E3 type. Now in this process, the final electron acceptor is the oxygen, and it will convert into the H2O. Okay, and oxygen is reduced into the form of water molecule. Now this up to this complex 4 processes, the main thing is that for complex 1, due to this shuffling of the electron around, due to the shuffling of this high energy electron word I am using, that due to the in complex 1 shifting of high energy electron, okay, shifting of this high energy electron, around 12 kilocalories are generated. 12 kilocalories generated. Okay, so this energy 12 kilocalories are generated in the complex 1. So this energy is utilized to throw the 4 protons from the matrix to the intermembrane space. Okay, so the 4 protons are thrown back to the intermembrane space. Now in the complex 3, around 10 kilocalories are generated. Okay, so the 4 protons are thrown back to the intermembrane space. But here in the complex 4, not too much energy is generated, only some energy, that energy is sufficient to throw only the 2 protons. Okay, in the complex 4, 2 protons are pumped into the intermembrane space. By throwing this 4 and 2 protons, the proton gradient is created. What is the proton gradient? That the intermembrane space become more positive and the matrix layer or matrix side is become less positive, means it become negative. This will form a proton gradient. This will form a proton gradient. Okay, so these protons will have to pumped back into the matrix because it will create a imbalance. So these protons have to the pump back into the matrix. So now the main difficulty is that the inner membrane is impermeable to the ions. Okay, so the inner membrane is impermeable to the ions. Okay, inner membrane is impermeable to the ions. Okay, impermeable to ions. So what will do? These things you have to keep, this question will raise in your mind. Then the proton channels will present. Now the complex fifth come into the action, that the ion channels which present in the complex 5. So these protons will just pump back into the matrix, and when through the ion channels, so these protons will pump back into the ions, into the matrix part. So during this process the ATP is convert into the ATP. Okay, but this process in the complex fifth process, the chemiosmosis process will take place. So that process we will study in the oxidative phosphorylation. So in the ETC cycle or ETC chain, you will write only that these protons will pump back through the ion channels which is present in the complex 5. So that process during this process the ATP is converted into the ATP. Okay, so this is whole for the electron transport chain. Now we will summarize what we have read. Okay, now the complexes in the electron transport chain, the target behind the electron transport chain is that to convert the NADH and FADH2 into the ATP form. There are the 4 complexes involved in this process and which are embedded into the inner membrane. Okay, so during this inner membrane complex 1, by just converging this process, the high electrons will throw back into the matrix, from the matrix into the inner membrane space. So proton gradient is created, and when the balance will reversed, okay, so when the proton will pump back into the matrix, the ATP synthase complex 5 just come into the action and convert into ADP into the ATP. This whole for the ETC cycle. Okay, so in the complex fifth process we will study into the oxidative phosphorylation. We will make the separate video for that. Okay, I hope you have enjoyed my lecture. Any doubt, kindly ask in the comment section. Otherwise, please like, share and subscribe. Thank you.